Merci beaucoup pour l'invitation. Merci Aurélie, merci Emmanuel et FILFOIE. Et c'est génial de venir à Paris, de retrouver des amis. Je suis très contente d'être là. Et je suis très impressionnée avec les FILFOIE et avec les réseaux que vous avez. J'ai lu aussi, parce qu'on n'a pas une structure comme ça en Espagne, et forcément, ça donne des idées. Je parlerai de la maladie à la mode, de la PSVD, mais je ferai la présentation en anglais parce que je ne suis pas sûre si je me ferai comprendre en français. Normalement, je parle qu'à la maison en petit comité, et ça c'est compliqué pour moi d'expliquer tout ça. Mais on peut essayer de faire la discussion en français après. En tout cas, je comprends très bien, meilleur que je parle. So changing into English, I'm gonna show you a little bit of the data that we have. This is very, very preliminary data that we have in PSVD and using a single cell transcriptomique. So I think that it's very clear that we are very motivated with this disease, and the reason is because we do not know anything, and we see the patients, and we do not have anything to offer them. So recently, a few years ago, we decided to change the name, and this is why we came with the porto-sinusoïdale vascular disorder terminology, because we have been calling the disease in different names in different regions of the world, and we decided to come with a common term that can be an umbrella term where we put things together, patients that they do behave in the same way but have been called with different names. And this was the main aim of finding a name that everybody can use and being able to gather big cohorts and learning from the technique. So it's a rare disease, usually affects young individuals with a little bit slight predominance in male, and most of the time we do the diagnosis with patients with already signs of portal hypertension or decompensation, even if it can be diagnosed in patients without portal hypertension, but we know that from recent studies performed here in France. Most of the time, also, these patients, they do associate other conditions, and we are still learning about the prognosis, but of course, having portal hypertension and associated disorders impair the outcome. I'm not going to go through the biopsy because Alexandra explained you very, very well the challenges and why we still need a liver biopsy to make the diagnosis, but in general, it's an amazing disease, and it gives us a big challenge because we do not know anything. We don't even know how to identify patients, and we still have a lot of patients with a misdiagnosis, not detected, not diagnosed in many regions of the world. We do not have biomarkers, and we cannot predict the outcome of the patients. We need an expert pathologist to make the diagnosis, and the only thing that we do once that we make the diagnosis is following the patient and applying symptomatic treatment, because we do not know what is the reason causing the disease, and we have no physiopathologically driven treatments yet. So the idea is can we apply omics and can we move? Because in the last years we have been learning a lot about the natural history and more in the clinical side, characterizing our cohorts, but we thought that it was time to move to a more mechanistic approach and trying to understand what happened in the liver of these patients. So the first studies that we start doing, and this is when you compare with other rare disorders, we are very, very, very at the beginning. So we started doing bulk RNA sequencing in the liver of the patients and trying to identify if this is a disease that is completely different from cirrhosis, and what are the pathways that we can target, or what are the things that are going on there. And we could see in this study a few years ago that yes, it has a transcriptomic profile that is completely different than the one that we see in patients with cirrhosis, and then there is a lot of homeostasis, oxidative stress, and lipid metabolism that it's altered in this population when we compare that with healthy volunteers and liver cirrhosis. Why then moving to single cell? I think that this is obvious. So the info that we have is like common things. Like we know that those are the pathways, but that may be explaining the disease, but we do not know what is the reason or what are the cells important to the development of the disease. Something that is very intriguing is that the disease is patchy, so we do not have an homogeneous distribution in the liver. So being able to compare the affected versus the non-affected regions is something that it's appealing to understand the challenge, and then trying to know what are the important cells, and especially the crosstalk and the driver, to start like really understanding what is causing the disease. And this is why we decided to start like doing all the sequencing, the single cell sequencing, trying to first come with an atlas, describe what is going on in the livers of these patients, and of course with the aim of trying to derive biomarkers and therapeutic targets, things that we can target. So we put together a proposal with all these groups that you see in the slide, and combining clinical expertise with translational and basic expertise. And we won this grant, and now we are like starting doing all these experiments that I will explain you in a while, and with the objective of really learning things. So we are participating in Spain, France, Italy, Germany, and Israel. And Germany and Israel are the ones leading the translational part. So far what we have is like we did single cell in 16 patients. So we did 4 control, 7 PSVD, and I will explain you why 5 cavernoma. Those are patients with thrombosis, but in normal liver. We have bulk RNA sequencing from a big cohort, the cohort that I showed you before coming from Barcelona, and new patients, so a total of 39 PSVDs. And then we analyze, having a control for PSVD patients is also complicated, so we have been also deciding or studying what is the best control. If having a control, a biopsy, that by definition we are doing a biopsy because we have a suspicion of something, or doing in the setting of a liver donor transplantation. And we also have cirrhosis as a control. And in the more recent stage, we have been doing also spatial transcriptomic, doing Visium HD in samples from Barcelona and from Paris. So as I told you, this is like very, very preliminary, but and we did that with transjugular liver biopsies. That was a big challenge as well, because the amount of tissue that you get, it's complicated. So it was a lot of processing and adjusting. But we are happy because now we have a cell atlas with good quality and a good number of cells that enables us to understand what are the populations there in the livers, and trying to really characterize well the vasculature. So we were like, as everyone does in single cell sequencing, and trying to validate with the atlas that are available out there, and trying to understand what are the specific markers associated with each cells, and also the transcriptional relationship between the cells and how close the cells are to each other in the PSVD livers. So in all patients that we process, the representation of all the cellular components was quite good, and we could use all the patients that we sequenced, and we have representation in all the cells from control and from disease patients. So the first thing that we did was comparing what we have in the bulk array and RNA and see if we could compare that with the single cell sequencing and trying to identify what are the specific things that we see in the PSBD. And as you can see in the graph, we came with a few markers that seems that are important for PSBD, and this is FAP4, PL-BAP, collagen 15A1, and mainly those. And those are expressed specifically in liver vascular endothelial cells. So we were like very happy that when we compare these patients with the controls of the patients with cirrhosis, there are like things that are mainly coming from the endothelium that are giving us that are really differentially expressed and highly expressed in PSBD patients. And then we have a map, and we have each cells expressing different cells. So now the game starts, and now we are like trying to understand what are the different things that we see in each different subtype of cells. And so we ask what are the cells that are affected, because we know that this is a vascular disease. We know that the vasculature is affected, but we do not know if it's primarily affected, if there are like other cells that are affecting the vasculature, or if the hepatocyte has something to do in the disease, because usually the liver parenchyma is kind of normal, or at least it's not similar to what we see in cirrhosis. And those are the number of differentially expressed genes that we have. So hepatocytes, they do suffer from the disease, and I think that this is also easy to think that when you have impairment in the vasculature, these hepatocytes, they also suffer, but we do not know yet, and we are like trying to analyze signals coming from the hepatocytes to see if they are only reactive and suffering from whatever is happening in the endothelium, or if they are like really contributing to the disease. But as first step, we decided to be focused on the liver vascular cells. So we took this vascular component, and then we tried to compare that with the atlas that are out there in the liver, in the normal liver and in the cirrhotic liver, to see if we could differentiate the same amount of vascular cells and the same subtypes. So when we apply what has been things that has been described previously regarding sonation and regarding differentiation between what is a central vein or a portal vein or an L-sec, periportal or pericentral, and also detecting arteries versus the veins, we can detect all these populations in our patients with PSBD, and we have representation of all the compartments, and we are like ready to go to do the examines, because we do have what has been described previously. And this is separating all the vascular type of cells, the portal vein, the periportals, the sinusoidal periportal, the sinusoidal pericentral, the central, and the artery. And the markers that I told you at the beginning that were like really up in patients with PSBD, those are interestingly seen mainly in portal vein or in periportal sinusoidal cells. So we think that this is the important region where a lot of things are happening, and this is also what the pathologists see in the livers. And they also see a lot of changes in the central sinusoidal space with sinusoidal dilatation, and we also have change there. We are still like trying to understand what is going on, but it seems that we can have, or everything makes sense from what we know from the pathology studies. So we started to trying to identify what are these cells expressing, and all these markers that are expressed in the portal vein regions, if they resemble to something else. And it seems that whatever happened to the vasculature in hepatocellular carcinoma, it's kind of similar to what we see in PSVD, and those are again the markers that comes all the time that were described also in the studies performed by different groups, including Valérie Paradis, showing these markers, and maybe the cells in PSVD resemble somehow this oncofetal reprogramming that happened also in HCC. So those are like the first sets of experiments, and what we are doing now is like trying to understand the communication between the cells, and now we are analyzing ligands, doing ligands and receptor analysis, trying to understand what are the signals and what are the communication more important between cells. We were like initially thinking that maybe the crosstalk with immune cells and the endothelium was the most important thing, but it seems that the myofibroblasts are more important, or the crosstalk between the myofibroblasts and the PSVD. So, but still processing and trying to understand what is going on there. And we are also doing BCMHD in blocks and in biopsies of liver tissue in patients with PSVD. So we had a first set of samples that, of course, didn't work well, and now we are repeating the experiments, and let's hope to have the results soon. So once that we had all this data, and then we ask ourselves if we can use that information to make something that in the clinic is sometimes difficult differential diagnosis, because patients with PSVD during follow-up, they do develop portal vein thrombosis. So sometimes the patient comes to the clinic with a portal vein thrombosis, and we do not know if there is like a normal liver with a thrombosis or a PSVD with a thrombosis. So we decided to also analyze via single cell a sequence in patients with cavernoma that was that appeared in a completely normal liver, and then we apply also the analysis, and then we will compare in PSVD versus control, cavernoma versus control, and PSVD versus cavernoma, trying to identify what is the things that are completely different, and looking at the analysis, asking a very simple questions like, are those the same things, or the signals that we see in cavernoma are the same as one that we see in PSVD, or those are like two different things? And even if they share a few things, there are things that are unique and maybe can help us in the future to have kind of a signature that can help us differentiating in patients with thrombosis if they have a PSVD or if they have just a pure cavernoma. And again, the markers that I told you before, the collagen 15A1 and the FAB4, also FAB5, and other markers seems that are differently expressed in patients with cavernoma versus with PSVD. So a lot of things to do, yet we are like very motivated with the progress of the study. So I hope that in 1 year we can have a better understanding of what is going on, but we have now starting to understand what is the molecular landscape, and I think that with all the information that we will generate, we will be able to have unique biomarkers for diagnosis, but also for prognosis, and because I didn't show you the Visium HD because it's very preliminary, but we can see that in the regions where we see more signals are the regions where we can see also the histological lesions, or where the pathologists detect the histological lesions. And I think this will be a nice way of understanding better the disease and a breakthrough in our understanding that hopefully end up in finding new therapeutic targets. So this is the RITA group. This is how the project is called, and we had a meeting a few weeks ago in Barcelona where we discussed all this data and more data. And thank you very much for your attention.